Right. So if it's all right with David, I'll just very quickly introduce him. Um, so David Evans is best known to most of us as the Apiarist due to his popular weekly blog at theapiarist.org. David is based in the Scottish Borders and he's an emeritus professor of virology in the School of Biology at St Andrews University, with interests, among others, in deformed wing virus and chronic bee paralysis virus. He says on his site, I used to be a scientist and a beekeeper, but now I'm just a beekeeper who talks and writes, sometimes about science. I promote local bees and honey and responsible, sustainable beekeeping. And tonight, David's going to talk to us about bigger queens, better queens. So, David, over to you.
Am I? I thought I was going to. I thought I was going to talk about Donald Rumsfeld and preparing for the season ahead. Right. Give me 1 second then. I will have to get my. I'll have to open another open another slide. I was goodness me. Right. Let me let me find the presentation. Right. It's a much more interesting talk, the Bigger Queens one. So you've made a wise choice. I apologize. Right. Here we go.
In our little preamble there, we failed to. I saw the picture of Donald Rumsfeld. I didn't realize that that was part of a presentation. I'm sorry.
Yeah, that was there we go. Okay. Right, I'm going to share my screen so you don't need to look at me any longer. And and there we go. All right. So yeah, so Donald Rumsfeld. So I give a talk on preparing for the season ahead, which is a fine presentation, I'm sure, but it's a bit dull for experienced beekeepers, and it sounds from the preamble there that a lot of you are reasonably experienced beekeepers. Um, and I talk about the unknown knowns, and that's why Donald Rumsfeld's there. So, okay, I'm going to talk to you, going to talk to you about something completely different now. Bigger queens, better queens. So there may be an odd hiccup now because I won't know what order the slides are coming in. So let's see what happens. Okay, so this is me. Angus has kindly introduced pretty much everything on here. I think the one thing I would say is that my main interest in beekeeping is queen rearing. And I've been queen rearing for pretty much all of the time I've been beekeeping. I learnt my queen rearing from Terry Clare, the late Terry Clare, way back at one of the spring conventions when the spring conventions were in Warwickshire, because that's where I did that's where I started my beekeeping. And I've always found queen rearing fascinating, and I think it's by far and away the best way to become a better beekeeper. I don't necessarily think I've become a better beekeeper, but I think for most people, it's the best way to learn how to become a better beekeeper. So I'm going to talk to you not about the mechanics of queen rearing. I'm going to talk to you much more about how the colony rears queens and then how we can use that information to rear queens and hopefully better queens and bigger queens for reasons you'll see in a second ourselves. So I did ask Angus when we were in the sort of preamble when we were having a chat earlier, what proportion of the association membership rears queens, and he didn't really know the number, but he knew that there were some very experienced beekeepers, and probably, I'm guessing, a lot of you don't kind of actively rear queens, by which I mean you might not do any grafting and things like that. And I hope I've structured the talk, or I believe I've structured this talk in a way that there will be something for both groups. There will be information there which, if you don't rear queens now, you'll think, but I can take advantage of what the bees do and rear good quality queens myself without doing all of that grafting palaver. And if you're an experienced beekeeper and grafting's no problem to you and you use mini nukes like they're going out of fashion, everything's very easy for you, then there's probably still some information here which will make you think about whether that's the very best way to rear queens. Anyhow, let's see if that bears out. So, who's the talk for? As I said, beekeepers with little or no experience. And the take-home message here is trust the bees. They know how to do it, and they will rear perfectly good queens if you let them get on with it. And I'm going to tell you about ways and methods in which you can let them get on with it. And there's potentially the case that grafted queens may actually be substandard in terms of in comparison to the queens that the colony rears themselves. But it's also, as I said, there's something in here, I hope at least for very experienced beekeepers, and it should make you think, are there better ways I could do this, taking advantage of what we know about how the colony rears queens? So that's the introduction. When does the colony rear queens? And you'll know some of this already. There we are. Okay, so you know that the colony will swarm. If a big strong colony, early midsummer, something early or midsummer, something like that, they'll produce new queen cells, and the old queen leaves with the prime swarm, and there may be casts produced afterwards. So that's a swarming response. Then there's a supersedure response. This is often late in the season. The queen is essentially replaced in situ. The old queen remains. They rear a new queen, having judged for whatever reason that the old queen isn't up to the job. And quite often they'll coexist in the same hive for a period, 1, 2, 3 weeks or months. I had a colony go into the winter, which had two queens in it, and it had two queens in it since the second week of May. And they're both marked, both clipped, both present, but we'll see who's left in the spring. This is typically a late season event, as I say, but the key point here is that the colony doesn't produce swarms. And then finally, there's the emergency response, and this is the way we rear queens. Almost always, this is the way we rear queens when we're actively involved in queen rearing. We generate a colony that is queenless, terminally queenless. It cannot rear a queen because there's no material suitable for it to rear a queen. And then we provide it with that material, like grafted larvae, and bingo, the colony rears new queens from that. And this is, so the emergency response is what happens when the colony is suddenly rendered queenless. And it's rather difficult to think of examples when that might happen naturally in a hive. The queen's living in a very protected environment, but they obviously have a means of doing it, and we take advantage of that when we rear queens. I should say that during the emergency response, it did say at the end of that last slide, if you, for example, take a colony and you just split it in two, and you have one half that's queen right and the other half that's queenless, the queenless half will raise new queens, but it may still swarm. And under the emergency response, the studies show that around about 50% of the colonies that are produced like that, just by splitting a colony and walking away, around about 50% to produce swarms. So if you think about the process of rearing queens, during the first 6 days of development, from the time the egg is laid until the time a larva is 3 days old, it is what's called pluripotent. And that means if you give it the right stuff, which in this case is royal jelly, you'll generate a queen. Whereas if you feed it standard brood food, standard bee food, then it will become just a worker. And the diet that larva is fed, because the egg isn't fed, but the diet the larva is fed is determined by the orientation of the cell. If the cell is pointing downwards, it's fed royal jelly. If the cell is pointing horizontally, it's fed normal food. So queen cells are vertical. And you know all of this stuff. Okay, so here's a diagram of that process, and you can see that that section that I've highlighted in yellow. That's the pluripotent period. That's the period when the fate of that developing bee is determined by its diet. And after that, whatever has been started will continue, if you see what I mean. So if it doesn't get any royal jelly in those first 3 days, then really it's not going to become a queen, and it'll end up being a worker. So the told you about the first bit. The virgin queen emerges 16 days after the egg is laid. She spends a couple of days maturing in the hive. She'll go on a few orientation flights, several orientation flights usually, and then she'll go on mating flights. Now, most beekeepers underestimate the number of mating flights queens take. It's not unusual for them to take 2 or 3 or 4 mating. I thought that was my doorbell. 2 or 3 or 4 mating flights. She then returns to the hive, has another day or so during which time she packs the spermatheca with sperm, and then she starts laying eggs. And she'll stay in the hive for essentially the rest of her life, unless, of course, she swarms. And you probably know all of that. So during the swarming impulse, the queen lays the eggs in those queen cups, and you'll probably be familiar with these. You've seen them. Sometimes we call them play cups and things like that. They're larger than a worker cell, and they're generally located towards the bottom of the comb. Not always, but generally. I think the suggestion is that the queen's footprint pheromones are they're less prevalent in that position of the comb, and that induces the production of these. And when the queen does lay in them, then they end up getting fed royal jelly, but they're oriented downwards. During the supersedure response, the queen again may lay eggs in those queen cups, But the colony may actually choose an egg that's been laid in worker comb, so horizontally oriented, and then re-engineer it to make it vertical. To complicate matters further, it's known, and studies have shown this pretty clearly, that the workers will move eggs from worker cells into queen cups and rear those as queens. And again, during the emergency response, the eggs can be moved by workers, but because the queen hasn't laid those eggs in queen cups, because the colony's been rendered queenless just like that, then the workers have to re-engineer the cell to make it point vertically downwards, so that the developing larva is fed royal jelly. And all of the importance of that will become apparent in a few minutes, or at least I hope it will. So here's a typical swarm cell. That queen's gone. You can see the cap of the cell is open. She's off. It's down at the bottom corner of the frame. Here's a typical supersedure cell, often located in the center of the frame. I wouldn't trust either the position or the number as being indicative of supersedure. If it were early in the season, I would be applying swarm control to this colony. But if it were late in the season, well, actually, probably the colony will have swarmed because that cell's sealed. But if it's late in the season, I would assume that that was a supersedure cell. And here are some emergency cells, and it's in a colony I'm queen rearing. So this box is actually queenless, and I've put a frame of young larvae up above the queen excluder there, and they've drawn out some of those cells as emergency queen cells next to my cell bar frame, which is the one with the writing on the top. So most of that stuff you'll know already. Anybody who's been keeping bees for 2 or 3 years will be familiar with that. But what I'm going to talk about really is what the colony does about it. So why are bigger queens better queens? Because we want to rear better queens for our beekeeping. Why are queens that are bigger better is a simple question. Given the choice, what does the colony use to rear queens from? I'm going to talk a little bit about drones and mating. I'm going to talk about what the colony uses, what the colony chooses, why they choose, does the colony choose specific eggs and larvae, or that it's just a free-for-all, that is take anything that's available. And I'm finally going to talk about how the mother queen influences the quality of her daughter queens specifically. Because knowing some of these things, I think it will be an encouragement for beginners to trust the bees a little bit more, because they know how to do this thing really well. And I think it's an opportunity for the experts to do some experiments effectively to test other methods of rearing queens, which might produce better quality queens. So what's the definition of a good queen? And I would, well, we can define them in any way we want. It's obviously the characteristics of the queen, the colony, the stability on the comb, their productivity, frugal with stores over winter, and a host of other things. But essentially what it comes down to is that good queens head strong, healthy, and productive colonies. And the characteristics of that colony are largely, not exclusively, but largely determined by the quality of the queen. And if you forget about beekeepers and beekeeping altogether, the reality is that natural selection has yielded queens that seem perfectly good at heading large colonies of bees and making lots of honey and things like that. And if we understand how the colony does it, I think it will help us do it. So here's a quote from a book that's what now? It's 60 years old, I guess. Yeah, Laidlaw and Eckhart. So their book on queen rearing, they have this quote that says, "The value of a queen depends upon her ability to lay sufficient eggs to maintain a strong colony appropriate for the various seasons of the year." And if you read that book, I've got a copy on the shelf somewhere behind me. If you read that book, you will recognize the methods that they use to rear queens are essentially identical to the methods we still use to rear queens now. So things haven't progressed in 60 years. And actually, the methods that Laidlaw and Eckhart used were probably developed a further 60 years before then. So why are bigger queens better? If you take a pluripotent larva and you feed it for longer, it will eat more royal jelly, and it will therefore have better nutrition. And because it's been fed more, it will be physically larger. And that's as simple as that, really. I mean, the earlier they start feeding that larva royal jelly, the more chance it will have to become a larger queen because it will just simply be fed more, get more visits from workers, and it will be fed more. And here's a very poor quality diagram illustrating that. And we've known about this for years. So Jerzy Wojk demonstrated this way back in the 1970s. He reared queens from old larvae and young larvae, and he demonstrated that the queens reared from older larvae, by which I mean 2 or 3 day old larvae, had reduced reproductive potential. That is, they had smaller ovarioles, they were physically smaller, they had a narrower thorax, for example, and they stored less sperm. And then 10 years after that or so, not just a reduced reproductive potential was demonstrated, but Nelson and Gary demonstrated that they actually weren't as good queens because they didn't produce as much brood. These older larvae didn't produce as much brood, and they didn't produce as much honey in comparison to younger queens. And here's a cartoon diagram showing that. So there's an egg. So it looks like an Easter egg in the top left-hand corner. If that egg is just fed standard brood food, it follows the blue horizontal line. But the more royal jelly it's fed, indicated by the thickness of the arrows, the larger the queen it will produce. And more recently, David Tarpey's done some excellent work in this area. David Tarpey, 15 years ago or so, compared queens that were reared from 0 day old larvae and 2 day old larvae. And I've marked where those are on this on my cartoon here. And he demonstrated, just to put some numbers on effectively those earlier studies, that queens from 2 day old larvae were physically lighter, 13% lighter. They had a narrower thorax, so they were smaller bees. They had a narrower spermatheca, so they could store less sperm, and they stored substantially less sperm, which means over the course of their lifetime, they'll lay fewer eggs. And not only do they lay fewer eggs over the course of their lifetime, but they physically lay smaller numbers of eggs anyhow, and so they produce 40% or so less workers. And actually, he went on to show also that quite interestingly, those queens mated with fewer drones. The older larvae queens mated with fewer drones. That means there's less genetic diversity in the hive, and I'll come on to why that's important a bit later on. And the mating number of drones later on. So, given the choice, what would the colony choose to rear a new queen from? And from what I've told you already, you would think they'd choose the youngest larva possible. But actually, in a colony, they can also choose an egg. What I mean is they can choose a 3 day old egg. And so right from the get-go, right from the moment that egg hatches, they can start feeding it royal jelly. And this graph shows, I'm not going to talk about how they did the experiment, but eggs that were 48 to 72 hours old are preferably what the colony chooses to rear queens from. Not larvae at all. They will choose an egg in preference to a larva. Now, almost all of the experiments I'm going to talk about where the scientists have studied the biology of queen rearing, they've studied it under the emergency response. Bear that in mind. And of course, that's the way we rear queens as well. So it's relevant. So colonies would choose 3 day old, given the choice, they'd choose 3 day old eggs. And it turns out that not all eggs are equivalent. Some eggs are physically larger than others. And if you look at the acceptance rate of larvae, the larvae that are most preferable to the colony are those that come from the largest eggs. And again, so this was another experiment that was done when they looked at larvae from eggs of different weights, and the colony would choose the largest eggs, the heaviest eggs, to rear their queens from. If the colony isn't given eggs to choose, if the colony is choosing larvae, and they might choose this if, for example, you're grafting larvae and adding grafted larvae into a colony, or if you provide a frame of eggs is perhaps a better choice. If you provide a frame of eggs and larvae into the colony, if they choose to use a larva, which larva do they choose? Now, at the very crudest level, there are two different types of larvae. There are larvae that have already been visited many, many times by workers and therefore very well fed. And there are larvae that have not been fed so well, so they are poorly nourished. And they recapitulated that using, as you can see in this image here, they recapitulated that by laying mesh over developing larvae and leaving that mesh in place for a few hours, 4 hours, I think it was. The mesh on the right-hand side is big enough to allow the workers through so they can continue to feed the larvae. These larvae are literally just hatched. And on the right-hand side, the mesh is too small for the bees to get through. So those larvae are effectively starved for a short period. They're starved for about 4 hours. Then they take the mesh off, they add the frame into a queenless colony, and they say, which does the colony choose? Which larvae would the colony prefer to work with? And on the right-hand side, it shows that they would choose the non-deprived larvae. That is the larvae that were fed throughout their development so far, just the first few hours of their development. They would choose non-deprived larvae over the deprived larvae. On the left-hand side, they took larvae from exactly that frame I just showed you, and they grafted them into queen cells and into plastic cells and presented them vertically, just as we would when we were rearing queens. And you can see there that they make no distinction between the non-deprived larvae and the deprived larvae. So that should make you think, when you're picking larvae, if you're doing grafting, when you're picking it, you don't know whether that larvae has been visited by lots of workers or not. And the colony would, given the choice, it would choose the ones that have been fed well. But by the time you've presented them vertically into a colony that's terminally queenless, they make no distinction and they'll work with whatever they can, whatever they have available. So bigger queens are better queens for the reasons I explained. And if you want to go and look at that material, David Tarpey's studies are some of the best. The youngest larvae that are less than 24 hours old, less than 12 hours old, produce the largest queens because they're physically fed more. And if the colony is given the choice, they'd choose old eggs rather than larvae. They'll choose larvae from the large eggs over larvae from smaller eggs. And if they're given larvae at all and the larvae are in horizontally presented comb, they'll choose well-fed larvae. So I'm going to take a slight detour now and talk about drones and mating. Genetic diversity increases colony fitness. This was a hypothesis that was kicking around many, many years ago. And what do I mean by colony fitness? It's essentially any characteristic that you could measure the colony's performance by. So, for example, the amount of comb it builds, how disease resistant it is, how much brood does it produce, and so on and so forth. Doesn't matter what you measure, it's a physical measure of the fitness of the colony. It turns out, cut a long story short, that colony fitness is determined by the number of drones the queen mates with. Essentially, it's a case of the more the merrier. The more drones the queen mates with, the fitter the colony is. And this was work that was done by Tom Seeley and Heather Matilla back in I don't know, quite remember, this is 2007, so 20 years ago. I'm going to show you one data slide from it. And just look at the lines that have the filled squares, the black coloured squares, or the open, the white coloured squares. Those numbers, the graph, what you're plotting there is over a period of several months during the year. That's the number of workers those colonies make. Now, what they were comparing here were queens that were instrumentally inseminated with sperm from a single drone or with sperm from 15 drones. And the black symbols, higher up the graph, therefore there's more of whatever it is they're measuring. The black symbols represent the number of workers that queens that were instrumentally inseminated with sperm from 15 drones produce, whereas the open white symbols are the number of workers that are produced by the singly inseminated queens from a single drone. So the diversity comes from multiple drones, and a colony with greater diversity, greater genetic diversity, produces more workers. And Seeley measured all sorts of other criteria and demonstrated that all of them correlated with higher genetic diversity gives higher colony fitness. So if 15 is better than 1, how many drones does the queen normally mate with? And often beekeepers would say 10 or 12 or 15 or something like that. And this is often referred to as the effective mating number. If the queen in that experiment I've just talked about mated with 15, would 30 not be better? And what about 60? And it turns out Keith Delaplane's done this experiment. He did it about a decade ago, again using the same method, instrumental insemination. And he showed that the more drone semen, the more drone choice, the variety of drone sperm, then the lower the mite levels were in the colony and the greater was the colony fitness. But the reality is that free living bees don't mate, free living queens don't mate with 60 drones. They probably mate with a lot more drones, as you'll see in a couple of minutes, than we think, but they probably don't mate with 60. And this 30 or 60 or 90 or whatever the number is, that's called hyperpolyandry. Polyandry is a female mating with multiple males, and hyperpolyandry is multiple males. And it turns out that free living bees are not what's called hyperpolyandrous. They don't mate with 60 drones. And probably that's because of the combined risks of predation. The longer they're flying, the more chance they get eaten by a swallow, and also sexually transmitted diseases, because deformed wing virus, for example, is transmitted when drones mate with queens, and that becomes a sexually transmitted disease. And there are others as well. So again, the more drones she mates with, the riskier it is. Just want to come back to this point about how many drones does the queen mate with. That it's If you look in at a frame of developing larvae, the larvae in adjacent cells have different fathers. They have the same mother, but they have different fathers. And the number of different fathers that contributed to that frame, they're called the patrilines. It's a line of descent from the male ancestor. And multiple patrilines coexist in the colony. The sperm, as I've said, is used randomly. Different cells adjacent to each other will have different fathers. I'm not going to talk about the mechanism for counting patrilines, but there are ways of doing it. You don't need to sample every larva. You can mathematically calculate the slope, if you like, of this curve by sampling a few, maybe a few hundred, but it tells you the number of patrilines in the colony. And the patrilines that are used for rearing queens are called royal patrilines. Now, we don't know what they are in advance, but the fact that they end up in queens subsequently makes them royal patrilines. So, given the choice, which patrilines does the colony choose to rear its queens from? And there would be an expectation. Oh, I should say one final thing. Yeah. If you looked at a frame of bees, a frame of larvae, there will be different numbers of larvae from each of the patrilines that contributed to that. So you might have 400 larvae from one particular father and only 130 from another and only 20 from another. Because the sperm is used randomly, there's going to be a right mishmash on every frame. So all other things being equal, if the colony had no preference for which patriline it used, it would choose, or you would expect queens to have, to be derived from the larvae which were most common in terms of patrilines on the frame. So are larvae chosen randomly? That is, from the most commonly present patrilines. So people have done these experiments, and I'm not going to show you any data. I'm going to show you my cartoon of the process. So here are 100 larvae, which I've color-coded, and there are 11 separate patrilines there. And I've given you an indication down the left-hand side of the diagram of the frequency, their percentage present. And the top four, the white, yellow, cyan, and whatever the next color is, I'm colorblind, green, I think. Those four represent about 60% of everything that's on the plate. And the others, kind of the also-rans, they're only a much smaller proportion. And some of them are very rare indeed. So if you present a colony with a frame like this, with a mixture of different patrilines, what do they choose? And it turns out they preferentially choose the rare ones. They don't exclusively choose the rare ones, but you will find the rare ones much more frequently than you would expect if you just pick them randomly. So rare patrilines are disproportionately represented in the queens that are reared from a colony. And this is an amazing experiment, frankly. But it does, and we don't know why it happens, but it definitely seems to happen. So we're on to the second part of the take-home messages. Colonies favour the rarest patrilines. So if we're thinking now about practical queen rearing, what have I told you so far? What bearing does that have on the way you might think about rearing queens? So the first thing is you should use larvae that are from colonies that are well fed. Goes without saying. You don't want to have poorly nourished larvae chosen by your colony that you're rearing queens in. Second thing is that the colony that is rearing the queens should itself be very well fed, because you want to make sure that they get as much royal jelly into them in that short period during which they're feeding. And the final thing is, when you're getting your queens mated, have them in areas where there are huge numbers of drones available for queen mating, because clearly large numbers of queens contribute to higher colony fitness, and they might also contribute a little bit in terms of generating a good range of patriline's for the colony to choose new queens from. So here's a few things to think about. Can you distinguish a 12-year-old larva, yeah, 12-year-old larva, 12-hour-old larva from one that's 24 or 36 hours old? Now, I could probably distinguish between a 12 and a 36. I might struggle between a 12 and a 24, and that's with strong glasses. How could I select larvae from the largest eggs? I have no information at all about the eggs. What about me as a queen rearer? What about me discriminating between the well-fed larvae in the colony and those that were nutritionally deprived because they haven't had enough visits from workers? Again, I haven't a chance of doing that. And patrilines, again, you can't tell which patriline a particular larva comes from unless you basically crush it up and do the genetics on it. So again, I have no opportunity of testing any of those things. But the bees do all of those things when they're rearing queens. All of those critical decisions are made when the larvae rear queens, and they know how to do it, and they've learnt why to do it, if you like, through evolution. Their survival depends upon it. And yet, as I said earlier on, Laidlaw and Eckhart, and going back to Doolittle and things like that, the methods we use now are the methods that have been used for the best part of a century. And they take no account of any of these things that we now know about how a colony rears queens. So if you're a beginner, as some of you probably will be, at least not in terms of beekeeping, but in terms of queen rearing, you're probably thinking, how on earth could I as a beekeeper make such good decisions as the bees are making? Why don't I just let the bees do it? And actually, I think you're right. And if you're an expert, you're thinking, and I was thinking when I read all of these papers, when I select larvae for grafting, when I sit there hunched in the front seat of my car over a frame of eggs and larvae, picking larvae up with my sharpened matchstick or whatever, I know nothing about where those eggs come from, their patriline, their nutritional status. What are the chances that I'm missing the best larvae? And the reality is the chances are pretty good that I'm missing the best larvae. Okay, so are all eggs the same? No, we know they're not all the same, because genetically I've already shown you eggs have different fathers. That represents different patrilines, and particular patrilines, the rare patrilines, appear to be chosen by the colony to rear queens from. I've already shown this bit, so I'm not going to talk about it before. But what about eggs that are laid in queen cells by the queen in comparison to eggs laid by the queen in worker cells? Are they the same? And it turns out they're not. And this brings me to the final bit of the science I'm going to talk about, which is what's called the maternal effect. And I need to explain what the maternal effect is and talk about how it influences the queens that a colony produces before talking about any ways that we might exploit it. And the maternal effect is something that was not first identified in honeybees. It was identified in other insects, and actually it exists in other species as well, ducks and fish and stuff like that. And it effectively says the environment of the mother, and it uses scientific terms here, but it influences the phenotype of the offspring. And the phenotype is essentially the appearance and function of the offspring. So in particular environments that the mother finds herself in, when she's laying eggs, she will put more into, for want of a better term, she'll put more into laying eggs that will be turned into offspring that will continue to propagate down subsequent colonies, for example, if it were an insect. And the maternal effect can be cover a variety of different functions or appearance characteristics, if you like, of the mother's progeny, but for example, size or sex or behavior, all sorts of different characteristics. It depends what the species is. But the take-home message, the important part of this, is that the offspring in which the mother puts more effort into, and I'll talk about that effort in a second, have increased fitness. They are better quality, whatever it is that she happens to be producing. And I'm talking about a bunch of sort of kind of arm waving-y type things here. I need to give some examples. But the question comes down to this last one here. Queens laying eggs in queen cells are in a different environment. The cell is oriented differently. It's sometimes in a different position in the frame, a host of other things. It's physically a different size. Do those eggs and the queens that are reared from those eggs differ from the queens that are reared from cells, from eggs that are laid in worker cells? And to cut a very long story short is they do. But I'll tell you a little bit more about the maternal effect on this last slide here. So here are 4 examples of the maternal effect. So, for example, one is that certain ants, I think it is, they lay eggs that develop into winged offspring, that is, offspring that can disperse in particularly crowded environments or when the environmental conditions are adverse. So normally they produce unwinged, flightless progeny, but the environment is judged by the queen to be adverse, to be not the sort of conditions that she would want to rear flightless youngsters, to rear flightless children in. And so she changes the type of eggs she lays. They develop into winged progeny and they can fly to new environments that are more conducive to the colony surviving. Some insects, when they lay eggs under conditions of nutritional deprivation, lay larger eggs. That means the egg has more nutritional resources to start with, which means it has a better start in life. Ducks and fish, female ducks and female fish, make a greater effort in egg production if they judge the male, the drake or whatever a male fish is called, if their perceived quality of the male that they mated with is a higher standard. And there's another example of ants there. In 3 of these examples, and it's useful to have them highlighted here, the queen lays larger eggs under particular conditions that are going to go on and continue the generation, if you like, contribute to the next generation. So that comes back to that question I asked a second ago. Do queens lay different sized eggs in a queen cell as opposed to a worker cell, honeybee queens? And if they do, if they do those eggs develop into larger queens? And if they do develop into larger queens, do they develop into better queens? So there's 3 sets of questions here. So the experiment was done by some Chinese scientists. There have been 3 or 4 papers produced on this in the last 5 years, I think. And the experiment is relatively straightforward. They 3D printed using, they 3D printed some comb, and the comb either had 4.9 millimeter diameter worker cells, which are shown in, I guess, B in the middle of this picture there, or they had 9.7 millimeter queen cups. So these were much larger, about twice the diameter. And this was a standard sheet of it's effectively a sheet of plastic foundation, and they put this in a frame, and they put it in a colony that was queen right, and the queen laid eggs in those plastic cells, and then they had a means of prizing the cell out of the sheet of foundation, the plastic foundation, and then presenting it vertically so that it was reared as a new queen, and that's shown in the bit at the bottom. And essentially, it's kind of like a custom-made Nico system or Agenta system, if you're familiar with those. I think I might have a picture of those a bit later on. This system has been described in the American Bee Journal back in 2019. And one of the great things about the American Bee Journal is all of the archives are available online, digital archives available online free. You don't need to be a subscriber. So you'll all be able to read that if you want it. So they got the queen to lay in these sheets of foundation. They prized the cups up. They presented them to a queen rearing colony, that is a queenless colony that was had lots of young bees in it, and then they tested the size and the quality of the resulting queens. I should say as well, they physically measured the size of the eggs, and the eggs were larger, but I'm going to skip over that bit. So cutting a long story short, the queens do lay larger eggs in the larger diameter queen cells. Those resulting queens that were reared from those larger eggs, were physically larger. They were heavier. They had a wider thorax. They had more ovarioles, and they laid more eggs. And in addition to that, they produced more brood. So they had all of the characteristics you might want in a bigger, better queen. They also had some evidence that the workers that were reared by that second generation of queens were themselves physically larger and collected more nectar and pollen. So it suggests that there's actually an improvement across the board. It's not just it's not just the queens themselves that are better, but it's the progeny of those queens that are better as well. There's relatively little data on that side of the thing, and I'm really just focusing on the queen for the moment. So these really are bigger and better queens. And I think there's quite significant implications for beekeeping here. If you're a beginner, the bees know exactly how to rear new queens, which includes selecting the right larvae and making all of those choices I talked about a minute ago. And I think it's just a case of trusting them. Um, and I'll show you a way in which you can do that. So if you provide a queenless colony, terminally queenless colony with a frame of eggs, they will choose the best larvae to work from, and they'll rear new queens from it. And so the process is really very, very straightforward, and you need almost no, you don't need any specialized resources for it. You don't need to do any grafting. You don't need to do any fancy manipulations of the hive. And I'll talk through it, and I've got a diagram to follow it. So this assumes you want to rear, let's say, 4 or 5 new queens. You've got a colony that you can use for queen rearing. The quality of that colony is immaterial. They can be stroppy bees, they could be not very productive bees, they could have all sorts of characteristics you don't like. What you need is a frame of eggs and larvae from a colony that has the characteristics you do want. And you might need to work with a friend in the association to get that. You know, if you've only got 3 colonies, it's rather difficult to judge which is the best and which is the worst. But if between you, 2 or 3 of you, you've got half a dozen, you've got 9 or 10 colonies, you can probably identify which colony is consistently a better performer, and you can use that as a source of eggs and larvae. So let's go back to this potentially stroppy colony or poorly productive colony. As long as it's a strong colony and as long as it's a healthy colony, it will do in terms of rearing a new queen. So you go to that colony, you take the old queen out, and you leave the colony for 7 days. And by leaving it for 7 days, there are then no more eggs or larvae that are pluripotent because they've all got too old to be reared as new queens. So you go through that colony and you remove all the queen cells from it. That colony is now terminally queenless. It cannot rear a queen to replace the one you took away 7 days ago. Push the remaining frames together, and that's going to be your cell raising colony. This colony is terminally queenless because of the way you need to inspect the frames to get rid of all of those queen cells. You really need to shake all the bees off every frame, and that's very disruptive to the colony, and it certainly doesn't leave it in a state that's sort of conducive to accepting new eggs and larvae the moment you put a new frame in. So leave that colony for 24 hours. And then into the middle of the colony, add a frame of eggs and larvae from a good donor colony. Just add it to the center of the brood box. And you must have eggs and you must have young larvae on that colony. It doesn't matter what else is on that colony, but eggs and larvae are critical. And 10 days later, that frame and that frame only will have queen cells on it. And you can leave one queen cell in the colony, but you can cut the other queen cells out and you can use them in other colonies to requeen them, to requeen other hives, and you will have reared queens, and you will have let the bees make all of those critical decisions I talked to you about early on. 2 weeks later, maybe 3 weeks later, depending on the weather, we always want to check too soon, but you check for mated laying queens in those colonies. So here's a cartoon of the process. You start with your queen right colony in the top left-hand corner, you take the queen out. You might put her into a nuke or you might cull her, depending on her quality and how much you want her really, and the colony will inevitably rear new queen cells and they'll be scattered all around the hive. So you go through that hive, you shake all the bees off every frame and you crush every queen cell. That colony is now terminally queenless and you leave it for 24 hours. And after 24 hours, in the bottom right-hand corner now of my diagram, you add into the center of the colony this frame you've chosen from your selected donor colony. Good quality bees, healthy, stable on the comb, highly productive, all of the things you want in your bees. You leave them for 10 days and they'll produce new queen cells on that frame. It's their only chance of survival and they will do it. You leave one queen cell in the colony, but you remove the others, cut them out with a good circle of comb around. It's a messy process because there's going to be other brood in that frame as well, but you need to take the queen cell with a good flanking chunk of comb and you put it into a queenless nuke, for example, and you wait for that queen to get mated. And that's queen rearing. To improve your chances of success, keep good records. You should always keep good records anyhow. Use your best quality colony for the source of those eggs and larvae. But the quality of the cell raiser, as I said, is irrelevant. But it must be strong because you want to make sure that those developing queens are fed really well. Feed everything. Feed the donor colony for the eggs and larvae before you take the frame out, and then feed the cell raising colony. It doesn't need much. 200 or 300 mils of syrup a day is fine. You must remove all of those queen cells after 7 days to render the colony queenless, and you need to shake all the bees off the frame. It's very important because otherwise they'll rear a queen from their own stock and your timing will be off. You won't know quite what's being produced when, and you won't have had any input into the selection of the quality of the bees in terms of the donor colony that they're going to make. Cut those queen cells out with ample flanking comb, and it will work. So here's a colony of mine that I'm doing this with. It's a reasonably strong colony. I've got a frame feeder in this colony. That second slot a little bit further up is a frame feeder for me to feed syrup to them because there was a bit of a nectar dearth when I was doing this. But you see, I've taken a frame out of the middle. I'm about to put a frame from a donor colony in there. So what are the advantages and disadvantages of this approach? So there's no manual selection of the larvae. This is a really distinct advantage, particularly if you're averse to grafting, if you don't like the idea of grafting, or you've tried it and not succeeded. There's no grafting. All of the manipulations are completely familiar to a beekeeper with even 1 year's experience. The bees are doing all of the selection and they're very, very good at it. You can improve your stock. Over 2 or 3 year period, you'll see an improvement in the quality of bees, assuming you choose good quality stock to provide those donor frames of eggs and larvae from. And it should produce some spare queen cells. But one of the problems is, and the disadvantages are in italics here, you don't have a huge amount of control over the number of queen cells they're going to produce. They might produce 3, they might produce 33. The more they produce, the more difficult they are to cut out without damaging some of the neighboring ones. So you just need to be a little bit careful there. But as a means of producing 3 or 4 or 5 or 6 queens, this works. It can be a little tricky, as I say, to cut out the other queen cells. And the precise timing of emergence can vary a day or two. You don't have quite as much control over the process or predictability of the process as you do if you were grafting larvae. But frankly, that shouldn't matter. It works well. So that was for beginners. What about experts? What about people who think nothing of grafting 50 larvae in an afternoon and raising 35 or 40 queens from those? You're familiar with the methods that you use to do that. You rear strong queens, or you rear queens by adding grafted larvae into a strong cell-raising colony, usually a queenless cell-raising colony. And those queens are good, or they're at least good enough. And you've been doing it like that for years. Beekeepers, as I said, have been doing it like that for a century. But could they be better? So are there ways in which we could exploit what we now know about some of the ways that the bees use to rear queens, potentially taking advantage again of the flexibility that some of those plastic queen cups, the Nico-like or the Genta-like systems offer, easy handling of the queen cells, predictable numbers and things like that, but that also take advantage of the maternal effect. And I don't know the answer to this, but I'm trying, and I know other beekeepers are trying this as well. So the first thing is that commercial queen cups, the ones that are sold by Genta and Nico and JZBZ, and if you're familiar with these names, you'll recognize them. They're the 3 things along the top. If you look at the sizes on my crummy set of micrometers, they're all too small. They're all at least smaller than the ones that were used in those studies demonstrating the maternal effect in honeybees. We don't actually know that they are too small. Okay, we just know that they are smaller than the cells that we used in the scientific papers on the subject. But the other thing is these plastic queen cups are designed to be put in one of these kind of boxes, for want of a better term, in the bottom left-hand corner there, which presents them as a little frame when you cage the queen in there, and the queen lays up the thing. But each of those boxes has an aperture above the larger diameter cup that the queen is going to be reared in that reduces the width of the orifice, that's the width across the top of the cell through which the queen lays an egg. So my strong suspicion is that the queen is going to lay worker eggs in these plastic cups because the environment she finds herself in is not going to be like that of a queen cell. It's going to be much more like that of a worker cell. Now, again, I'm guessing here, but I think that's probably a reasonable assumption. So I think it's time to start getting creative. So I've been using and other people I know who are doing this, we've been using 3D printers to print plastic queen cups to present to a colony. And those plastic queen cups are physically larger than the ones you can commercially buy. So the idea is that you would add a queen onto a frame where you had these larger diameter plastic queen cups. The queen would lay in those cells, and then you'd present the resulting larvae in a standard system, a queenless cell raiser, vertically presenting them, and they would raise them as new queens. So the advantage here would be there's no grafting involved because the queen's doing the work in putting the egg into the queen cup. But in addition, it would exploit our current understanding and knowledge of the maternal effect in honeybees. Of course, formally, we'd still, or whoever does this and gets it to work properly, will still need to demonstrate that the resulting queens are bigger and better. But certainly based on the science that's been published, that would be the expectation. So this is very much a work in progress. I've got one additional slide, which I've realized I haven't got in this talk, but I'll mention at the end. This is very much a work in progress. These are some plastic queen cups that have been printed. The one right in the center, facing upwards, is a clone of a Nico cup, and that's the same diameter as a Nico cup. The other ones are larger, and I've made a variety of sizes, but up to about 9.7, 9.8 millimeters in diameter. The other thing I've done, and you might be able to tell from the ones in the top left-hand corner there, they've got a thicker upper rim. And there's a separate set of studies have demonstrated that the acceptance rate for larvae, grafted larvae presented to a cell raising colony, the acceptance rate is influenced by the thickness of the rim of the plastic cell cup. And the thicker the rim of the plastic cell cup, the higher the acceptance rate. And this was a study that was published 5 or 6 years ago or something like that, I think. So I've printed some which are both larger diameter and have thicker rims. Now these are 3D printed out of the material you buy to running your 3D printer. I knew nothing about 3D printing before I started this, I should add. And I had no information about whether that plastic was toxic to bees or whether they would be repelled by it or anything. So one of the first things I had to do was to simply take the plastic cell cups that I'd made, graft larvae into them in a normal way, and then see if they would rear queens. And they do. And you can see there's 4 different cell cups there. There are 4 different types of material, and they're rearing queens in all of them. So I'm not worried that there's any toxicity issues here. Then it's a case of, actually, I'll show you both these last pictures. Then it's a case of presenting an array of cell cups and putting a queen in there. So I built cages for the queens, cell cups shown there on the bottom right-hand corner, and I cage the queen in there, put her under the queen excluder, leave her in the box for 24 hours. And then I go back and harvest, for want of a better term, the cups with eggs in them. And this is where the first of the major problems occurs, and that is because the queen doesn't lay very many eggs. She does lay eggs in these cups, these oversized cups, these larger cups, but she doesn't lay very many eggs. And I really, to get this to be widely accepted, the queen's going to need to lay more eggs. And I've talked to the Chinese authors about their study, and they had exactly the same experience. They find the queen lays relatively few eggs in those cups that I showed you the photographs of earlier. So, sorry, let's go back to this one here. The queen lays rather few eggs in this. And when you take the cups, there's still an egg in there. And you really need to get those eggs to hatch into larvae. If you present eggs to a cell raising colony, they usually eat the eggs, whereas they don't usually eat larvae. So you need to incubate those eggs for a day or two before you eggs in the cell cups for a day or two before you take them out and present them to a cell raising colony. I've got eggs in there. I've raised one or two queens. What I haven't got is enough eggs to get any sort of meaningful numbers out of queens at the end, or to sort of optimize any of the process. And you'll appreciate working in southern Scotland is probably not the best place to try and do these things. Um, Yes, I'll talk about the other thing right at the very end. So the choice that the colony, this is a summary now, the choice that the colony makes in terms of the larvae it chooses to rear as new queens is probably the most important decision that colony will make. And the bees are exceptionally good at doing it. They've been around for millions and millions of years, and they know how to do it. And yet, most of the ways we rear queens ignore all of that accumulated knowledge, for want of a better term. And that choice is made upon the choice is made upon a variety of things that we now know about, and probably a host of things that we don't know about. But for example, the age of the egg or the larva, its nutritional status, the patry line, and so on and so forth. And if you're a beginner, you can exploit all of that knowledge, and you will rear perfectly good queens using effectively the bee's expertise to do it. And you don't need to use any high-tech grafting approaches or anything like that. It just works. But for an expert, or more experienced beekeepers at least, we know how to graft and we can routinely produce queens like that, but we may well be missing the best queens because we're not taking advantage of the maternal effect. And if we can exploit the maternal effect by getting the queen to do the work and lay the eggs in the cup, and those eggs are physically larger, and as has been shown, they produce bigger queens, then actually if we can reproducibly and routinely do this, it will have all the advantages of the Nico-like system, and yet take advantage of some of this biology that we now know about for the colony. And of course, the other thing is that the technology for getting that second set of, that second point to work is also usable by less experienced beekeepers, because again, there's no grafting and things like that involved. So I think it's got a lot to offer beekeepers. I'm going to mention one final thing, which relates to the size of the box that the queen is in and the size of the eggs that the queen lays. And I don't have a slide of this, I'm afraid, though I do on the talk that I thought I was going to give. There's been some recent studies which show that the physical size of the box that the queen is in is inversely proportional to the size of eggs that she lays. And so in a large hive, she tends to lay smaller eggs, whereas in a very small hive, like a Keeler or an Apodea or something like that, she lays larger eggs. And the expectation would be, though it hasn't yet been demonstrated, the expectation would be that the small eggs that the queen lays, the large eggs that the queen lays in a very small box should produce larger quality queens, larger sized queens, which will be better quality queens at the end of it. That hasn't been formally demonstrated, but it's definitely worth investigating. Anyway, I've written all sorts of things about this under a set of titles called Bigger Queens, Better Queens, and talking about the size of eggs that queens lay in various environments and things like that, all on the website. And I would encourage you to have a look at that if you're interested. Here's one of mine going back in the box. Grafted, perfectly average sized queen, perfectly good colony, nothing wrong with it at all. But I think there are other ways of doing it. And knowing a bit about the biology of bees, I think suggests there may be some useful improvements that we should be able to make to our queen rearing. And even if they don't produce better quality queens, it's interesting anyhow, knowing how the bees do it all. So thank you for your attention. Fractionally over time, I apologize for that. And I hope there are some questions, and I'll try and answer them. Thank you.
That's brilliant. Thank you. Thank you so much, David. Anybody got any questions they'd like to ask coming out of that? Probably people with more experience at queen rearing than I have. Simon.
Um, One question, Dave. Really interesting talk, fascinating stuff. I will definitely play around with some of those ideas. Is there a risk when you put a frame of mixed brood into your queen raising colony that they build on older larvae and
I
produce
don't.
what we would call a scrub queen
Yeah,
or a
so
you know.
I don't think there is. I mean, certainly if you look at the studies done when you present a range of eggs and larvae to a colony, they're very selective in that they choose old eggs. A few of them will be produced from slightly older larvae and things like that. But that graph was pretty clear cut. They choose old eggs or young larvae. So I'm never worried about the colony making the wrong choice there. I have a strong tendency to always trust the bees. I know they get it wrong sometimes, but no, I don't think they should produce. If now, let me just qualify that. If the colony has no choice in that you only present it with older larvae, 3-day old larvae, then yes, I think absolutely they'll produce a scrub queen there. And that's because they have no other option. And it's their only chance of survival is to get anything they can out of it. But I think if you give them a range of larvae, they will choose the youngest to work with, or even the oldest eggs in the colony. Eggs tend to get eaten when you put them in when you put them in a foreign colony. So I'm not quite sure whether that's the best thing. But certainly I think they will normally choose the youngest larvae possible. Yeah.
Thank you.
Okay.
Mm.
Somebody with their hand up, I don't know what your name is. You seem to be called MJC iPad.
Yeah, sorry, my name's Matt.
Hi, Matt.
You're right. I was wondering, would you not be tempted to try and do a demo ray split and then just use the frames out of the top box effectively?
Yeah, so I'm not a big fan of Demarays for a variety of reasons. One is the season up here is a bit short. It might work fine for you, but our beekeeping season is 6 weeks shorter than yours, and it's yet one more thing to try and squeeze in before the rain starts again.
Mm.
Um, so yes, I mean a Demaray should work, and it should take advantage of all those things. Of course, doing a Demaray doesn't necessarily allow you, or doesn't, the way we've talked about it, or the way it is normally performed, it doesn't allow you the chance to improve your stock. Whereas the method I talked about, you using a different set of eggs and larvae from a good donor colony. But yes, I mean, there's nothing wrong with a Demaray. I don't like it because of the shortness of the season and the what happens to the drones, because you find loads of your drones get stuck in the queen excluder, don't they? Yeah. So it's not a method I'm keen on. Um, and the fact that it doesn't allow you to select and improve your stock, I think, is probably a drawback as well. But in terms of the quality of the queens from what you're presenting them, yes, it should be fine.
Cool. Because yeah, I was
Okay.
thinking effectively I'd take your best colony, do a demo ray on that, and then take a frame
Yeah.
out, reach the top. So you treat that as your cell raiser.
Yeah. So that should work. Yeah, that should be fine. Yeah.
Brilliant. Thank you.
Okay. All right.
Anybody else? Anybody got any questions they'd like to ask? Colin, you're muted at the moment, Colin.
I'm just wondering about the quality of the many queens that are generated in a swarm, because the bees and the queen have both had a chance to produce a better new generation of queens. Is that necessarily true?
Um, so so sorry, I'm not entirely sure I follow the question. So the queens that are generated when a colony is swarming, are they, is your suggestion that they're worse or better?
I'm suggesting they should fit all your criteria for good queens,
Oh,
better
I,
queens.
no, I agree. I absolutely agree. Yes. I mean, I know some people are anti using swarm cells because they think it's going to propagate swarminess and things like that. I've not seen evidence for that, but I haven't looked all that hard, I have to say. But you're absolutely right. Yes, a colony under swarm conditions will be clearly selecting the very best eggs
Mm.
or larvae to propagate itself, to reproduce. And so, yes, they should be, absolutely, they should be the best ones they can produce. Yes. Yeah.
Okay, yeah.
Can I ask
Matt,
another one?
yeah, go ahead, Matt.
I was going to say, is there anything research you would say on mating nukes as well? So would you prefer, therefore, to move a frame or a single queen cell into a 5-frame nuke or a 2-frame nuke? Because I know you've written on 2-frame nukes, or would you use a queen mating nuke? Is there actually a difference in the quality of the queen there as well?
No, I'm not aware of any work that's been done there. I think there is a difference for the beekeeper though, in that, in my view, uh, I, as I said right in my introduction, I think queen rearing is one of the best ways to learn about beekeeping, and I think that a lot of beginners are put off queen rearing because of all the fiddly bits, if you like, lots of fiddly stages, each of which might not work very well. And one of the fiddly stages that doesn't work very well is the management of mating nukes. Those small mating nukes, I think, are kind of difficult to manage. Um, I don't think there's any data on the quality of the queen that results from being mated from one of those nukes. I'd say so. I favor 5-frame nukes for two reasons. One is I find them easier to manage. Secondly, if I'm teaching beekeeping, I find it a lot easier for the trainees to learn using a 5-frame nuke than a mini nuke and all that entails. Um, but the one thing I'd say in favor of mini nukes is that the queen gets mated appreciably faster than in a larger box. And if you've used mini nukes, you're probably familiar with that. Certainly, it's the queen's out and back before you know it, and she's laying very quickly usually. Whereas in a big hive, for example, you can wait ages for the queen to mate. And I've talked to other beekeepers, some very experienced beekeepers who've seen exactly the same thing. I don't understand why that is. The final thing I'd say about mini nukes is I find it impossible to judge the quality of the queen in a mini nuke. You just can't do it on a 3-inch square lump of frame. She needs to be on a proper size frame to see how good she is. And that again means I end up putting her in a 5-frame nuke anyhow. So I get them mated from there anyhow. But I'm not aware of any influence on the mating itself. No.
Thank you.
Okay.
Any other questions? I think we've exhausted the questions. Last chance. Going, gone. No, I think that's it. David, thank you so much. It was absolutely fascinating, and there's a lot to go away and think about there. So that's what more could we ask? Thank you so much.
You're very welcome. And I'm pleased you told me which talk I was supposed to give before.
Yeah, that was
before I
lucky,
launched
wasn't it?
Oh dear,
very
yeah.
good.
I'll just yeah. Anyway, that's good. Yeah. So the thing I mentioned right at the very end about the size of the box and the eggs the queens lay, I've written about that relatively recently, and it's a
Ah,
fascinating story. And I think there are ways that again that's exploitable for beginners. So it's yet another thing. It's just the biology of bees is fantastic, and we don't take enough advantage of it. We started to learn these things, and we should take more notice of it and take advantage of it. Anyway, I hope you all have a great season
thank
and
you. You too.
better season than last year, perhaps.
Yes,
And
indeed. I
yeah,
was I was saying what a miserable time some of us had with losses, but
yeah.
so let's hope it's a better one this time.
Okay. Well, good luck. And even if it's a bad season, or rather, even if the losses are higher than they should be, you now have some idea about ways in which you can make good,
Indeed.
improve your stocks. Okay.
Indeed. Thank you so much.
All
Thank
right.
you.
Cheers now.
Bye
Bye,
now.
everybody.
Bye,
Bye.
everybody.